This Week in Parasitism is brought to you by the American Society for Microbiology at microbeworld.org/TWiP. This Week in Parasitism, episode number 16, recorded on September 8th, 2010. Hi, everyone. It's Vincent Racaniello, and I give you Dickson Despommier.
Hey, Vince. How you doing?
I'm well.
Good. Me
okay,
too.
That's it.
Yep. Are there any questions?
You're well. It is September. The summer is over.
No, the September 21st is the end of summer.
Yeah, officially,
Sorry.
but
I know,
culturally, it's over here in the United States.
In the United States, it's over. This is true.
It is time
to go back to work.
School begins. Well, we are working all summer, but school begins. So
yes.
Psychologically, you're starting anew in the fall.
Yep.
You have some projects you're starting in this fall?
Well, I've got projects that are ongoing that I'm continuing.
I know your
through
book
the fall.
Is going to be released this fall.
It is.
Correct?
It's kind of exciting, yeah.
October.
October 13th. That's
wonderful.
Right. Yeah. I'm happy.
And you're going to write another one, and another.
if they let me. It depends on what the people say about book number 1, I think.
It's going to be a blockbuster, I guarantee it.
Vince, I sincerely hope you're right. And so does the publisher, of course.
And you know that once you're rich and famous, you won't talk to me anymore. That'll be
Vince,
the end of it.
I'll either be rich or famous, but probably not both. So either way, I'll still talk with you.
You're already famous.
You're my friend, and I don't abandon friends.
You don't?
Do. No, neither do you. I know you
Excellent.
don't.
I only have a few, so if I abandoned them, it would be a problem. Just kidding.
Dear.
Last time we talked about trypanosomes.
We did.
We are still doing the protozoans.
Yes, there are so many of them.
And today, which protozoans do we encounter?
Well, Vince, today we're going to group them together into a syndrome. Rather than talk about them as individuals, we're going to talk about them as entities that cause diarrheal disease.
So the syndrome is diarrhea.
It is.
And
it's multiple forms. There's more than one kind of diarrhea.
Really?
That's right.
I have known only one.
Well, I bet you knew more than one, but you just didn't know you were knowing more than one.
Etiologically, most of the diarrhea that I would know would be viral etiology.
Such as?
Norovirus, rotavirus, and a few others, but those are the two biggies.
Yep, I would agree with you there.
And then I guess the protozoan diarrheas are significant also then, right?
Well, we have some bacterial ones that we didn't discuss just now.
I don't know which bacteria cause diarrhea. E. coli, of course. The pathogenic E. coli, Salmonella.
Shigella.
I'm sorry. Am I speaking correctly saying that the pathogenic E. coli will be causing diarrhea? Or is that
absolutely
incorrect?
No, that's absolutely right. Absolutely. It's called traveler's diarrhea because you exchange E. coli wherever you go, and the enterotoxin is slightly different no matter where you are, and that causes the diarrhea. So diarrhea is a condition that I'm sure everybody that's listening to this podcast has experienced at one time or another in their lives. And of course, every time you do get diarrheal disease, or infection or syndrome, depending on which ones those are caused by. Sometimes diarrhea can be caused by a toxin rather than an organism, as in the case of cholera.
Right.
Okay, so the cholera toxin, not the cholera organism, causes the diarrheal disease. And we know a lot about the mechanism that is engaged through the cholera toxin molecule, which, by the way, was derived from an ancient E. coli encounter and then altered along the way by some virus which gave it from E. coli to the cholera organism.
Right.
Somewhere in the Bay of Bengal, deep into the reaches of the...
Don't you teach in your medical ecology course about how you can predict cholera by the algal blooms?
Absolutely.
Is that correct?
Yep, it is. We'll get to that. Perhaps we'll have This Week in Bacteriology, and then I can talk about that sometime.
Yeah, that's right. But we could have This Week in Medical Ecology.
We could do that. We could have This Week in the Life of.
Right.
But today we're going to concentrate on 3 protozoans that are linked by common clinical features, and that the clinician has to struggle with in order to tell them apart because the treatments are all different. So if you made the diagnosis of, let's say, the first organism, which would be Giardia lamblia.
Giardia lamblia.
Right? That organism causes diarrheal disease. Okay?
Mm-hmm.
And so if you were to guess correctly that your patient had diarrheal disease caused by Giardia, then if you gave the proper treatment, which was metronidazole, which inhibits an anaerobic pathway, you could get rid of the organism. And lo and behold, the diarrheal disease will go away. But if it's caused by Cryptosporidium hominis, which is another protozoan that we can catch from either ourselves by cross-contamination with feces or by animal sources, then Vince is reaching for some paper so he can start writing these things down. Everybody listening. So we've got 2 organisms already. Now we've got Giardia lamblia and we've got Cryptosporidium hominis.
Hominis.
Right? They used to call it Cryptosporidium parvum, but they discovered through genetic analysis that there are several species out there.
In your book, it's called parvum.
I know. It's an old book.
Yeah, old in science.
2005.
Yeah.
We've got to bring this up to date, and we will, by the way.
Hominis meaning humans, right?
That's right.
But parvum meaning...
Good question, Vince. I'm afraid we're going to have to look that one up.
Unless
somebody out there...
There's something here called the internet.
That's true.
Parvum.
Parvum. I think it refers to its generic distribution because it can infect a lot of different mammals and it can also infect birds. So Cryptosporidium is a very unusual protozoan.
Well, all the definitions are Cryptosporidium parvum.
Exactly.
So I don't know what it means.
All right. Maybe it means something like ubiquitously distributed or something. Maybe we have a Greek scholar out there that's listening that can help us out here. But I claim ignorance as to know what the word parvum actually means. But
here we go. Web definitions for parvum.
Go on.
Frog. Oh my gosh. I'm not getting anything. Allium parvum.
Well, this is probably
species of wild onion.
It's probably why I don't know, because I've never heard anyone tell me or read anything that says parvum. Oh, yes, that means no. I don't know the answer to that. There's plenty of things I don't know the answer to.
Merriam-Webster says the word you've entered isn't in the dictionary.
Well, then they're out of date too. They need to upgrade their dictionary as I need to upgrade my book. All right.
Cryptosporidium parvum
and hominis.
And hominis. Same thing, no?
No, not the same thing. Genetically distinct.
All right. And then the third organism?
The third organism, Vince, is a conundrum.
It's conundrum C.
No. It's called Entamoeba histolytica. Histolytica. Now, each one of these organisms...
They're all parasitic protozoans,
They're all parasitic protozoans. They all live in the gut tract.
They live in the gut tract.
They do.
And we will find out who is the definitive host and the accidental host and the reservoir.
If there are any sexual stages or not, depending on the species in question. Some of these protozoans don't have sexual stages, as we'll see. So let's begin with just each individual organism group first to describe who they are and where they're found and who might catch them.
I have so many questions, Dickson.
I'm sure. And I have fewer answers than you do questions.
You have a lot of answers.
Yeah, some of them are even correct. But let me just start by saying that Giardia lamblia has been known since microscopy has been known.
I'll bet I know when that began.
Tell me, Vince.
A Dutchman.
Yes.
Named Anton Leeuwenhoek. Is that how you
Leeuwenhoek.
Leeuwenhoek.
Anton Leeuwenhoek. He wasn't the inventor of the microscope, but he was the progenitor of the microscope. He popularized the use of the microscope.
He was a lens maker.
He was a lens maker. This is true.
For glasses?
Yes. And
telescopes.
You know what's unusual about this is that his original microscope is on display in a museum in Leiden, Holland. And you can actually go there and visit the museum and see his microscope.
Have you?
I have not, unfortunately. I would love to do that, though, because a picture of his microscope can be found in the back of my book, which I have paid homage to Leeuwenhoek by actually showing a picture of him looking through his microscope. And you can see it right there on page 323, I believe it is.
Anton van Leeuwenhoek.
Anton van Leeuwenhoek. So,
microscopist extraordinaire, discoverer of the trophozoid of Giardia lamblia.
That's right.
I guess that's what the picture is.
I've superimposed him on top of a of a photomicrograph of Giardia.
Gee, did you use Photoshop for that?
I did actually, Vince. Thank you. The point of that image is that here is Anton van Leeuwenhoek looking at a specimen of his own stool. Now,
dear.
Now, Leeuwenhoek, being one of the first users of the microscope. Now, let's say that you were an inventor of the microscope, Vincent, and this is now back in the 1600s, and you had the choice of anything around you to look at. Would you gravitate towards stool material?
No, I would start with saliva. I'd prick my
a
finger
raindrop,
and get some blood.
Sure, or some dirt, or maybe a piece of paper, or, let's just look at whatever's available.
I know he looked at his sperm.
Eventually.
I wouldn't know how to do that.
We're not going to that subject. This is not This Week in.
But, you could go in the streams and look to see what's there.
And in the canals. And in the rain gutters, which he did.
Wine, beer.
Yeah, keep going. You can
food.
do it. Eventually, he ran out of the usual suspects to study under the microscope. And he ended up looking at a small fraction of his own stool, in which he saw these little animalcules floating about.
And they're moving.
They are moving. So this is a flagellated protozoan. So the trypanosomes were flagellated protozoans.
Correct.
The Leishmania, in part of their life cycles, were flagellated.
When they need to move, yeah.
Correct. And so what we have here is another example of a mastigophora. This is an entire phylum of organisms called mastigophora. Okay. So here we are.
What's the other group? The
Apicomplexa.
Apicomplexa.
Well, that's the second organism, Cryptosporidium.
Okay. No flagella.
None.
All right.
No apparent means of locomotion.
So Leeuwenhoek did this in roughly what year?
Back in the 1600s.
And he saw this, it's sort of a droplet-shaped organism.
Teardrop. It looks like a teardrop.
With a flagellum.
That's right. And, of course, he didn't have anything to stain these things with. He was just looking at the live organisms.
Now, why did he have these in his feces? Was he sick?
We're going to get to that. We're just definitely.
If I looked in mine right now, would I find some?
I don't know, Vince. How you been feeling lately?
I feel good. I feel very good.
Have you lost a lot of weight lately?
No,
have you been feeling lethargic?
In fact, I gained a few pounds.
Well, in that case, you probably don't have Giardia. So at any rate, it was discovered. It was one of the first organisms ever discovered because
Did he name it? Leeuwenhoek?
That's a very good question. Did he name it? No, it was named by two other people. Lambl, L-A-M-B-L.
Lamblia came from that.
That's right. And the word Giardia, I think, means something else. But again, you're pressing me up against the limits of my knowledge. Other people know the answers to these questions. And in medical school, I rarely had to answer that kind of question. So I don't know what the word Giardia means. But Lambl, it was certainly named after an investigator who took it up from where Leeuwenhoek left off.
So shortly afterwards?
Not so shortly afterwards. I think he was in the 1800s at this point.
But Leeuwenhoek didn't make any connection with disease, right?
Correct.
Okay.
This was during the early discovery days of microbes and disease. So the connection was loose, if you'll excuse the expression.
Stool, huh?
No pun intended there whatsoever. So, but in the meantime, of course, as the organism came under scrutiny from science, particularly from infectious disease people, the entire life cycle was revealed. And it's a very simple life cycle, very simple. You ingest an environmentally resistant stage called a cyst. It then goes down into your small intestine. And the cyst receives outside chemical information. The main thing it receives is bile, because you've probably ingested this along with either water or food, probably food. And so the cyst now, along with the bile salts, job in this case is to dissolve away a lipid layer on the outside of the cyst. And allow information now to go inside the cyst to tell it where it is.
It's always a cyst that you ingest.
Ingest. From this particular parasite, yes.
That's a cyst there in figure 1.2.
That's right. In fact, all the three organisms that I'm going to talk about have cyst stages or resistant stages in the environment. So that's what makes them so universally distributed.
So this goes into your stomach and begins to encounter bile? No, in the
small
duodenum, yeah.
In the duodenum, that's right. And so at the moment the bile salts start to dissolve away a lipid layer on the outside of this cyst, the pH of the environment becomes known to the organism.
Okay.
And as a result of that, it triggers the hatching response. And the cyst contains 4 nuclei and lots of inclusion bodies, okay, of different names. They've got fancy names like parabasal bodies and those are things, and axostyles.
Despommier bodies.
No, none of those. They're not there. So the bottom line, no pun intended on that either, is that the cyst, once having been now revealed to the environment of the small intestine, divides into 2. And each one of those transforms into a flagellated adult, which you could call the trophozoite stage. Okay? The lab technologist would call that the troph stage. They just abbreviate it.
So
troph.
1 cyst, 2 trophs.
Correct.
All right.
Correct. And that's a good ratio for this parasite to maintain because it gives it twice as much chance of succeeding.
But you said there were 4 nuclei.
There are.
Does each troph have 2 nuclei?
Yes. Yes. And now the interesting part of this is now that we can look back in time genetically and find out who begat who, so to speak. It turns out that Giardia is a pretty ancient organism.
How do we know this?
Well, we know it by the molecular clock that we can use to time things with. The rate of change of DNA is pretty constant over time, and if we look back in time to see how much change has occurred, we can now calculate how old this organism is. The other thing we can do is look at the ribosomal RNA configurations to find out what they best represent. And there are some archaebacterial-like sequences in this organism that actually says that this is related to the ancient life forms that gave rise to all the other life forms, supposedly.
This has been around for billions of years?
Apparently so, but how could that be, Vince? It's a parasite.
And it has to have something to parasitize. Yeah.
That's right. So there were no gut tracts back then. So how could
it was very different, right?
Well, you know what? It's funny because a lot of these might be facultatively adapted to life inside of a host rather than living outside.
Sure. Well, you can imagine at one time it was outside a host.
You could.
And then it found a host and said, I like this.
Right. And the host didn't say, I don't like you. So the next thing you know, you had a relationship.
It evolved. And now it has to be there.
Apparently
it's
so.
Lost its ability to be independent.
That's right. Well, you can culture these in vitro.
You can.
You can, yeah.
You can culture the trophozoites.
You can. In fact, you can even get them to insist now.
So these now emerge in the gut.
Right.
And
then what?
What happens next?
Exactly. It's an exciting story.
I bet they don't get into your blood.
You bet you're correct. This is not a tissue parasite.
It stays in the lumen.
Well, it doesn't exactly do that either, Vince.
Embeds into the wall.
It encounters the microvillar surface of the columnar epithelium, and guess what? It looks almost like a clownfish nestling into a sea anemone. I'm not kidding, because on the next page, we've got a picture of it right there to show you what a biopsy from a mouse with its own Giardia murus species. By the way, they're species-specific Giardia. That doesn't mean, for instance, that you could only catch Giardia derived from humans, because dogs and beavers and other wild animals, including deer, can also acquire Giardia lamblia, and they can then transmit it back to us, and bears also.
So I have questions, but you want to wait about that
transmission? I don't
know.
What's your question?
Well, I could see getting it from a beaver if you drink stream water somewhere, right?
Correct.
And same with other animals. But how do you get from human to human? Contaminated food?
Of course. I mean, just like you would catch salmonella or shigella or rotavirus. All of those are human fecal contamination events that you have to live with, unfortunately, because we live in groups.
So the person who handled the food that you ate that was contaminated was shedding it, didn't wash their hands properly. Were they ill, or are there people who have this and shed it?
We'll get to that.
Okay, sorry.
We'll definitely get to that. But just to complete the life cycle, the trophozoite stage then finds its way below the perimucosal layer. What's a perimucosal layer? Perimucosal layer is the mucus layer that coats the outside of the gut tract, below which there's this physiological space between that, which was secreted by the goblet cells, by the way, and the microvillar surface of the columnar epithelium itself. And there's a special physiology associated with the perimucosal space, which parasites, many of them, not all, occupy. And Giardia is one of those parasites that sits on the microvillar surface, sort of pushes them aside, and with its little suction cup-like organelle, it's It's not, it's a single cell, so it has no organs,
Mm-hmm.
but it has sort of a cup-like shape to it. It's like a sucker disc, okay, like a remora would suck onto the bottom of a shark. The same thing occurs when Giardia lamblia attaches itself to the gut tract. And it sits on the gut in very close opposition to the membranes.
Mm-hmm.
Now, one of these organisms isn't going to mean anything to a host, but
Sure.
this thing goes crazy after that and absorbs your pre-digested food. So here you are eating away, and all of the enzymes are working. The small molecules are going across the perimucosal space to try to get transported into the blood, and instead it counters Giardia. Okay. Now there's another feature of the gut tract that you have to be aware of. There's something called a lacteal. You know what a lacteal is, Vince?
No, I've heard of it though.
A lacteal is a little pore that sits at the platform of the villus pocket, and it actually absorbs fats. So all of the fats that you digest partially down into triglycerides, or from triglycerides rather into the fatty acid chains, those fatty acid chains are absorbed through the lacteal. And then they get involved into the lymph. And that's why when you eat a fatty meal, and if you could then cannulate the thoracic duct, which is where all the lymph eventually drains into the aorta or into the returning vena cava, then you would find it becomes a milky-like substance because of all the fat that's involved in this, right? Giardia prevents that from happening. Giardia plugs up that hole.
Lacteal.
Yep. And the fats are not absorbed as a result. So guess what the consequence of a heavy infection?
One Giardia is not
Who cares? Who cares, right?
One is not going to do anything.
One, two won't do anything.
You need
4.
thousands. You need millions.
Wait, I'm working up to something here, Vince. How about 8?
Where have these come from?
How about 16?
We ate a few cysts, right?
We did.
We didn't eat billions of cysts, right?
It could happen, unfortunately, but usually not. Usually it's a couple.
So in a mouse, you could probably give them a few cysts and they would become infected.
They've done that infection rate.
So do the trophozoites multiply?
They do.
And what do you get? More trophozoites? So
yes.
They just divide.
They divide and divide and divide.
And each has 2 nuclei also.
They divide and conquer. What do they end up doing? They end up coating the entire surface of the small intestine.
No, I don't think so. The entire surface? The small intestine is about 20 feet long, right?
No kidding. So this is the upper portion of the duodenum and jejunum. Just in that area alone.
They cover it, eh?
They cover it. So what happens to the fat?
It doesn't get in.
And then what happens to it?
The Giardia take it.
No.
Or it goes through.
It goes through.
It makes diarrhea.
And no, not yet.
It sounds like a fatty diarrhea.
It will be a steatorrhea.
Steatorrhea, that's right.
It's called steatorrhea.
Smelly, huh?
Because of the stearates that are generated by digesting fatty.
So the Giardia do not want this fatty material. It
Apparently
passes
not.
through.
That's right. Or they prevent the fat from being absorbed.
Yeah, but then in the
they might take up a little.
But in the lower intestine, there are no Giardia, so it should be absorbed there.
Actually, that's where your microbes are, right? That's where they are. They are the ones that prevent you from excruciating bowel movements. What they do is reduce all of the cellulose portion of your diet to them instead. What would you rather do, Vince?
Yeah, I say it would be huge otherwise, like the notobiotic
like
mice,
the germ-free mice. That's correct. That's correct. So these bacteria rarely get an opportunity to feast on fat. Rarely.
Ah.
But when they do,
they make methane.
They go crazy. They just don't make methane, hydrogen sulfide. They make a whole bunch of other things. Foul-smelling, fetid. They call it fetid.
Fetid diarrhea?
Fetid, foul-smelling steatorrhea.
I don't understand where the diarrhea or steatorrhea comes from because...
it prevents absorption of nutrients also.
The Giardia is preventing?
Yes.
Now, as you know, in the large intestine, you absorb water, and that forms the stool, no?
Right. Yeah, it actually takes water away from the bulk of material that's down in the lower gut tract, and that condenses to the form stool. That's correct.
So why is that prevented in Giardia?
It's not prevented. Well, it selects for different kinds of microbes.
Oh, okay.
All right. These are anaerobic bacteria in the large intestine that go crazy as a result of a renewed diet where fat is the major ingredient. So this has consequences for the host and for the bacteria. The host, without being able to absorb fats, what do you think happens?
Well, it becomes malnourished, right? Weak.
You become thinner.
Yeah, but not in the course of a few days, Dickson.
No. This is not in the course of a few days. This becomes a long-term chronic infection usually.
Really?
Yep, really, because in the beginning it's not perceived as an infection.
Oh, I see.
And until it becomes clinical, you might have a little bout of diarrhea disease, but to speak of, most people don't complain of this for at least a week to 2 weeks after they become infected, and then it starts to go into these chronic phases of it vacillates between steatorrhea and constipation of all things.
Mm-hmm.
It's got this weird diphasic clinical presentation, which confuses the heck out of most basic clinicians because a lot of them have not studied giardiasis, as the disease is called.
You taught this to medical students because they would encounter it, right? In this country?
Did. Well, let me tell you how many people encountered it last year. It's not a reportable disease, and yet the CDC has at least 20,000 cases on their record from last year alone. They just published their findings in the PubMed. So 20,000 cases last year, as opposed to 16,000 cases the year before that. Well, 20,000, Vince, I would add 2 more zeros to this one.
Probably. And does it kill you?
Of course not. No. It's a chronic long-term diarrheal disease which can weaken you. And if you're a kid, of course, and you're beginning your normal growth rate, it can actually stunt growth. But we'll get to that because there's some interesting findings with regards to Giardia.
The animals that have this, like the beavers in your picture there,
yeah.
Are they sick or are they chronically infected?
Chronically infected.
And they're fine?
Well, just like we are. Sometimes you're fine and sometimes you're not fine. You ask a beaver how they're feeling. He says, today I had a good day. I had a couple of logs. I slept in the hut. The wife was fine. The kids were not sleeping very well, though they were a little bit restless.
What if you give it to a mouse in the lab? Do they get steatorrhea?
They do.
Alternating with constipation?
They do, actually.
And they can live for long periods that way?
They can. They can, as long as their diet contains enough linoleic and linoic acid to compensate for their required fatty acids. So here we have this organism which Leeuwenhoek had it, obviously, because he found it in his own stool. And in fact, all of his mates that were also looking through microscopes that he made for them, they would have parties and sit around and look at stuff and all talk about what they were seeing. It was a hanging drop technique that they were using, by the way, and that's by candlelight. Can you imagine how crude and primitive that was? But,
Quite.
And they were still fascinated by what they saw. Because no matter how finely you divide life, you can see something else. All right, so this fed into the local concept that was developing also about the atomic theory of matter itself. Here's an instrument that you can use to see smaller and smaller and smaller things. Now, I wonder how small you can make a microscope to look at things with. And now we have an atomic force microscope. We can actually look at individual atoms. How remarkable we've come. And in just a very short period of time. We can look at the very structure and substance of the universe if we want. But it began with microscopes and Leeuwenhoek and Giardia. So this kick-started the entire quest of humankind to look as hard as they can, as finely as they can, at life in general. So how remarkable.
It's amazing.
How remarkable. So back to life cycle. This organism doesn't divide forever and take over your entire gut tract. There are some limits. Of course, everything has limits, right?
Not me.
Oh, well, okay. Vince is an exception, everybody.
We all have limits.
So the point is that eventually your immune system starts to say, wait a minute here. This is not a good thing.
We have a gut immune system.
We do. It's driven by some wonderful local immune response genes, which produce an immunoglobulin type A, which is secretory immunoglobulin type A. And we call it secretory because there's a little protein called peace protein. It's actually stuck on to the Fc fragment of the IgA molecules, and you get a dimer produced, which can't go back in the gut once it goes out. And it's extruded from the gut tract, and the peace protein is added, and it sits there in the mucus, sort of like a shield.
Yeah, that's important for preventing many gut infections like polio.
Indeed it is.
Rotaviruses.
And rhinoviruses and elephant viruses and ostrich viruses.
So these start to be made and they impact the Giardia.
They do.
What do? They reduce the numbers?
They interfere with this protozoan's ability to carry out its life cycle, but I'm not sure if we know exactly what it does to them. What it does, of course, is limit the number of trophozoites that can exist in the gut tract.
But
And eventually,
doesn't eliminate them.
In some cases it does, of course, because everything is self-limiting unless you die. So unless you go into form the chronic stage of the infection.
So you can die from this.
No, I was just saying that for other reasons.
Okay.
Like for salmonella, for instance. But for this infection, I don't know of anybody who's ever been diagnosed clinically with lethal Giardia. Even children that get it and become dehydrated die from something else, not from the organism itself. Okay, you die with it, not from it. That's the point. Although some pediatricians might disagree with that. And if you do, that's fine too, because I think we have a right to believe that this organism, under certain circumstances, probably could prove lethal for a newborn.
Mm-hmm.
Okay. Mm-hmm. But for most people with an intact immune system, even a partially intact immune system, we can bring it into limits. And that's true for most of these infections, by the way. So here we have this organism happily dividing away, preventing us from absorbing all kinds of things. Our villi respond accordingly by flattening.
Mm.
Now, when you flatten a villus, you've decreased the absorption surface. Okay, so not only can we block the lacteal, but we can also flatten the villus, and we've cut down on the absorption surface by a tremendous amount. So again, we're not absorbing the things that we should absorb, and it's passing down into our lower intestine where it's, the microbes have their way with it, but it's not doing us any good at all.
These guys don't live in the large intestine.
No,
right?
they don't. No, they don't. So you might want to ask, well, how do these guys carry out their life cycle? Because if you look inside them using the electron microscope, you won't find any mitochondria. There are none.
Is that why they're a parasite?
No. There's lots of things out there.
They make their own energy?
Well, they do, but they don't do it via the electron transport systems that exist inside a classical mitochondrion.
Okay.
So they're anaerobes.
Yeah, there are many ways to make energy.
That's right.
Yeah.
Well, this organism uses lots and lots of glucose because it is an anaerobe, and yet it's highly motile. So it's got high energetics, but it has no internal mechanism for converting and making a more efficient use of the glucose. So it just keeps churning out lots of product based on its anaerobic metabolism.
Is it obligate anaerobe?
It's an obligate anaerobe.
Mm-hmm.
It is. In fact, if you put it in vitro and try to ask it to grow, and it will, under anaerobic conditions, the moment you start to add oxygen, it slows down.
Yeah.
So then there are some other factors involved in this. I think it's population density that probably triggers the next response of this organism, and that is that a few trophozoites begin to round up and encyst. And as they do so, they divide their nuclei.
I see. So then we end up with 4.
4 of them. Now, there was a wonderful investigator here at Columbia, Sylvia LeBlanc, who I knew very well.
I remember her, Sylvia.
Sylvia, she worked with Lex van der Poel, in fact. And she was very much involved in Giardia research and was the discoverer of the fact that Giardia's nuclei, both of them, are transcriptionally active. Mm-hmm. So it's not just one nucleus. Both are making messages of various kinds. The whole time that this organism is busy dividing and filling up the gut tract, right? So it's interesting how she started in working on Giardia. It's a nice story because she actually began in Israel, working with a guy by the name of Richard Dickelbaum, who is in charge of our nutrition center here at Columbia now. Richard is a clinician, trained in Montreal, but resettled in Israel, in Tel Aviv, and began his career as a pediatric gastroenterologist. And he got interested in nutrition because he noticed that a number of children, regardless of their ethnicity, were malnourished, underweight for their age, and began to investigate what might be causing this, and latched on to the fact that Giardia was a common feature in those kids. So he began to study daycare center kids, in various places throughout Israel. Now, I know this story pretty well because Richard and I are good friends. And he tells his story multiple times in various ways as the data improved the story, of course. So what he eventually found out were the 3 kinds of kids, all of whom had... Don't correct me on these things, please.
I didn't. You corrected yourself.
I knew it didn't sound right the moment it came out.
All of whom.
So you'll all forgive me. I've been away on vacation for a while, so I'm busy.
No, you haven't.
Yes, I have. I just got back from Linz.
You were working.
I gave an hour lecture. The rest of the time it was piece of cake. In fact, it was a Linz-adored piece of cake, but we'll get to that later. There are three kinds of kids. One variety of chilled, quite literally. They had no signs or symptoms whatsoever from the infection.
They were infected, though.
They were infected. Another group had signs and symptoms, and of course they were infected. And then there was another group that was not infected. And what he did was he was very clever in noticing that there were, in every situation in the daycare centers, regardless of the close contact of these kids with each other, there were some kids in each of these groups that didn't have the infection whatsoever. So they served as the normal controls.
Mm-hmm.
And what he did was he measured their growth rates. It was very simple. He just measured weight gain over time. And in 6 months, you can get a pretty good curve for little kids as they start to grow because they're almost like in a log phase growth, right? So guess what he found, Vince? He made a graph, and then he starts out with the normals, right? The non-infected kids, and he grows the graph this way. Then he takes the kids that were symptomatic, and he shows their growth curves, and of course they were below normal because they're losing weight because this organism prevents absorption of fats, right? It's the third group that created the conundrum. So the third group, these are the infected but asymptomatic kids. They grew better than the non-infected kids. Better.
Wow.
Now, that's what he said too. This must be a mistake. They mixed up the numbers or something. Let's do this one again. Okay, so they moved it to a different daycare center, and they did the study again. They got the same result. Now, they didn't mix the numbers up. They said, I wonder how generalized this is. So they moved it to 3 other daycare centers, and they found for all 5 daycare centers in various parts of Israel, and keep in mind this was during some tumultuous times in Israel, which they've never had non-tumultuous times, but this was particularly a bad time for Hamas and other terrorist organizations to make life very uncomfortable for the citizens of Israel. These studies were being conducted under very unusual circumstances, and Sylvie LeBlanc was part of that group. And indeed, in all 5 cases, with regards to Giardia infections, you had 3 kinds of kids. You had the normals that grew normally, the kids with symptoms that grew abnormally low, and the kids that were asymptomatic that grew abnormally high. So apparently there's some genetics in this, right? So it could either be the genetics of the organism.
Or the host or both.
Or the host or both, right? And so far they haven't found out which one it is.
Do we know how it even works?
We do not.
Now, these kids must not be losing their fats, right? Otherwise they wouldn't grow.
Correct.
So they are asymptomatic because somehow they have dealt with what the organism is doing.
They have a genotype which allows them to remain in harmony with this now not parasite, symbiont, which gives them an advantage.
It's providing nutrients.
Now, wait a minute. If it gives the host the advantage, wouldn't this also be giving the parasite the advantage, Vince? This allows the parasite to be distributed all over the place without affecting the health of that particular group of hosts.
You would think that parasite would predominate eventually, right?
And believe me, it does. I mean, Giardia is found everywhere.
It's a really successful parasite?
It's extremely successful.
More so than...
No, there's none more successful than toxoplasma. I knew you were going to say that, but...
Toxo is 1. What's number 2?
Well, I think Trichomonas might be number 2, actually, which we haven't talked about yet.
Giardia
is up there with the best. I mean, it's one of the best protozoan infections in the world in terms of ubiquitous.
So do I understand correctly? You're saying that the incidence of infections without symptoms with Giardia is increasing globally?
No, I didn't say that. No, I never said that. I'm just saying that if you were a parasite and you wanted to distribute yourself throughout the world, the best way to do this would be to at least infect some groups that don't suffer from you and serve as a vehicle for the spread of this infection for other people.
Well, if it doesn't kill you, it's a successful strategy anyway.
Yeah, that's true. But, I mean, when you're sick, you don't feel like traveling much, right?
Yeah, but you just don't have to travel far to infect people.
No, you don't. That's true. But this parasite apparently is found throughout the world.
So we have no understanding of why these kids live peacefully with the parasite, nor how it makes them grow better. We don't understand the basis for that yet.
Don't have a clue, but if we could find that out, of course, we might unlock a secret to better living.
It's like a growth hormone analog.
A little bit.
So
could be.
How much bigger are these kids? What percentage?
I don't know, but it's statistically significant, so I can't tell you exactly how much. But, I mean, it's not 100% growth
Because as you know,
different.
Dickson, with cows treated with bovine growth hormone.
Oh, they grow much better.
It gets in the milk.
Yep.
Kids are bigger these days because
that's right.
That's
true.
That.
This is all true.
But it's not huge. It's not double.
No, it's not double. It's not anywhere near that. So, in the moment you stop drinking milk, of course, then it starts shrinking.
So the moment this is cleared, and has that been done, you clear the infection with a drug, do those kids stop growing faster?
That's a good question, because can you clear these infections with a drug? That's number one. And number two, if they're not sick, why would you give them the drug? So you have to be sick from something to give a drug to somebody.
Yeah, well, these kids were discovered to be positive because they're part of a study, so they might be treated. But normally, if you don't have symptoms, you're not going to get it, unless Leeuwenhoek found these and said, I don't want these. Take a drug.
Oh, Leeuwenhoek probably didn't want what he had, because I'm pretty sure he was suffering from the infection. But, like I said, it's a chronic intermittent infection in people. So,
but here in the hospital, you would be treated for it, right?
If you were diagnosed with it, and if you were sick from it.
How do you diagnose it?
Do it like Leeuwenhoek.
Look under the microscope?
Of course. That's the gold standard for all of these microbes, is that you look under the microscope.
Take a little bit of stool and you look at it.
Not just a little. You can take a lot here.
When you were a protozoal technician.
Or a parasite technician, yes.
Sorry, parasitic technician. Right. You were parasitic.
That's true. I was getting paid.
Did you look for Giardia?
Of course.
And did you find it?
Absolutely. All the time. And we used to throw up our hands in the air. Hooray, we found another Giardia.
You could look at it and say, there's a Giardia trophozoite.
A big deal. Oh, come on. Show me something I don't know.
And then the physician would treat.
Yep.
With metronidazole?
Metronidazole is the drug of choice today, but there are others. I mean, that's called flagell. Remember the name flagell? That was a European name for this drug. It was called flagell because it treats flagellates.
Including trichomonas, I bet.
You're right. Absolutely.
When we do trichomonas, I'll tell you stories.
Okay. I hope they're good stories, Vince. I enjoy a good story.
They're okay.
All right. So I haven't completed the life cycle.
Okay, that's fine.
So, we're back in the gut tract now, and we've now reached maximum number of trophozoites. At that point, there's some feedback to the population, and they begin to insist. Not insist, insist.
Are you saying there's some quorum sensing?
Vince, it's funny you should mention that, because the current president of the American Society for Microbiologists
Bonnie
is Bonnie Bassler, who was a champion for the quorum sensing molecule theory of microbial populations versus turning on those populations to do something.
Right.
So the answer is there are quorum sensing molecules for all of these things, I'm pretty sure. So we don't know what they are, but it's too bad, because if we did know what they were, we could prevent this organism from insisting.
And then it wouldn't be able to spread, because it is the cysts that infect a new host.
Vince, you nailed it.
Dickson, if you feed mice trophozoites,
they destroy them in their gut tract. In the stomach, they're dissolved.
They're not infectious.
Nope, not at all.
But if you feed them
cysts,
quadrinucleate cysts.
Thank you. Vince is busy reading on.
It's even harder than virology.
No, it isn't. No.
Much more complicated.
Can never. No, but this is a macrological approach. Yours is a micrological approach with the
molecular.
That's right.
So those are the only forms that will infect you. So if you could prevent them from forming. But Dickson, it sounds to me like this is not such a serious disease that anyone is really going to work on that.
You know what? It's true that there are some people out there that get paid to work on this. Fran Gillen is one of those people. Out at UC Davis. UC San Diego State University, and done some wonderful work on the curative powers of mother's milk, for instance, to actually lyse the trophozoites in a small infant's gut tract so that they don't become infected in an early age, and have a little bit of a chance to grow before they get infected from outside influences. So it's a fascinating infection. I think that the two nuclei being transcriptionally active is one thing. The anaerobic metabolism, all the pathways that are involved in this are very interesting as well. And there are a lot of people that would be very interested to know more about Giardia. In fact, I knew Don Lindmark, who was working in Christian Dedoux's laboratory the same time that I was there, and Don worked on Giardia and ended up at Cleveland State University and did some wonderful work on it. So It remains a parasite that has not gone away. This is a parasite that will probably never go away because for the very reason that Vince just mentioned, is that it doesn't cause a serious enough disease constellation of syndromes to warrant severe medical attention from the funding agencies such as NIH. Yeah, you have a question.
Yeah, I have a question.
Another question.
All right. So I'm under the impression that this disease or the infection by Giardia is relatively newly discovered. It seems to be on the increase in the U.S. Is that incorrect?
No, I wouldn't say that. I mean,
I have this recollection that so when I was a graduate student, I remember the chair of our department, Edwin Kilbourne, a very well-known influenza virologist,
indeed,
went to the Soviet Union and came back with an infection with Giardia.
But of course.
And this was really not known here in the U.S., not as well known as it is now.
It wasn't advertised, but
he let us know that he picked up an exotic infection there. No.
That's because he's a virologist. For him,
so if
it's not a virus, it's exotic.
If you go camping, is this a reason not to drink stream water?
It's called beaver fever. They call it beaver fever out west. Everybody knows it.
Do fish get it?
No.
Why not?
Because they're not warm-blooded.
Why do you have to be warm-blooded?
Their body, because that's one of the environmental cues that this organism uses to recognize where it is.
Interesting.
Yeah.
So how do we prevent it? Should we worry about it? And if so, how do we prevent it?
Preventing infection with Giardia is simple. Just prevent exposure to human feces. Good luck on that.
Or don't drink stream water.
Or don't drink stream water. So that raises a very interesting question, Vince. When you go camping, and you've done this, I know, because you are a Boy Scout master. Were you a master?
Leader. I'm a leader.
Right? So you get out to the campsite and you recommend to the kids, you got to boil your water. No,
we don't boil water anymore.
But you could.
We filter it.
What do you filter it with?
A filter. It's a commercial filter you buy. You have a hose.
All right.
There are two kinds.
Is it a ceramic filter?
You can have a gravity filter. No, it's not ceramic. It's a gravity filter where you do it overnight. You have a container. You put some stream water in, and it filters through a I don't know what the pore size is of the filter, and then it comes out the bottom. And there's some also organic removing.
It would be important to know what the pore size is, by the way, because...
So I did that 2 summers ago. I did a 5-day canoe trip in upstate New York.
Oh, yeah.
And we couldn't carry our water with us.
Through the Racket Lakes.
Racket Lake, and Long Lake, Blue Lake, all those in the Adirondacks. And we filtered our water every night. We would stop, and we had a pump as one kind, or the gravity.
Right.
So the first night we set up the gravity. We hung a bag on a tree, filled it with water from the stream, and then we put the tube into a gallon container on the floor. We went to sleep, and in the morning we woke up and it had tipped over and drained out. We thought a bear had done it, but in fact, when it got full, it just tipped over. So instead, we took the pump, and you pump it.
Yeah.
And that works well, except very quickly the pump gets clogged.
Doesn't it, though?
You have to open it up and clean off the gunk.
That's right.
But you get very clean water. None of us ever got sick.
Right.
And the cool thing is, from lake to lake, the taste is different depending on what kind of runoff goes into it. And it has different colors, too.
Yes. But I would venture to speculate that if you were to drink the lake water, by canoeing out to the middle of the lake, scooping up a big bag of water and bringing it back, it would be perfectly fine.
Is that right?
Yeah, it is fine.
Why is that?
That's right.
Why is that?
Well, think about it. If this is a beaver...
The beavers don't go in the middle of the lake?
They certainly don't.
I wouldn't know that, but...
Well, in those big lakes that you were on, Long Lake and Racket Lake, those are huge lakes, and the solution to pollution is dilution.
Who said that?
I don't know, but I continue to repeat it.
I've heard that before.
It's true. So the number of cysts that you might encounter in a liter of water near a beaver hut might be millions. But if you go out into the middle of that lake, or the pond even, and scoop it right out of the surface water, not down in the muck, down in the surface water.
Sure.
You probably wouldn't even have one.
It's an interesting point. I bet you're right.
Well, let me tell you why I know I'm right, Vince. Because what do you think New York City drinks?
Reservoir water.
It's not filtered.
Sure, they can't, yeah.
It's not filtered.
But beavers don't go into the reservoirs?
So
it's diluted.
I want you to do the following experiment, Vince, because this is a great experiment to do, and we're at a perfect time of the year for this experiment. I know a lot about, well, I wouldn't say I know a lot about, I shouldn't say I know a lot about, I don't know a lot about anything, but I do know about the ecology of lakes. A little bit.
Dickson, you wrote a book, or you wrote several books, so you must know something.
I must know something. Well, I had a lot of co-authors. Come on. I was
sure.
just the editor. So a lake in the springtime undergoes something called a turn. It turns. Okay, the lake turns. It transforms.
Sure.
It's the wintertime with the ice on the top. This is northern lakes, and the ice melts. The sun shines. The algae grow in the surface of the lake, and pretty soon you get a temperature inversion. Okay, as the 4-degree water sinks to the bottom, it stirs up the bottom and stirs up the muck on the bottom, which mobilizes all of the nutrients, and then it jump-starts the process of putting the algae at the surface and getting the lake to start to behave like a summertime lake. Well, in the fall, when the temperature cools down, the reverse happens. All right, so as that happens, the organisms start to stir up the lake, and the temperature zones in the lake disappear. You measure it at the top, in the middle, at the bottom, it's all the same. Now's the time. And the New York City reservoir system is undergoing this change as we speak. So what it does is it puts a lot of things into the water column. Now, if New York City water is not filtered, then it would be an easy experiment to actually go into the lab right now, Vince, turn on the faucet, fill up a 10-liter flask, and filter it through a 0.45 micron filter. Guess what color the filter will be?
Green.
Correct.
If you could get it through it.
Correct.
It would probably clog up pretty quickly.
Correct. So then you can take this filter and you can scrape some stuff off and put it under the microscope, and what do you suppose you're going to find?
Giardia?
No.
Algae.
Yeah? What else?
Protozoans.
Yep. What else?
Those... what do you... Oh, wait. The
I'm not going to
tip of my tongue.
I'm not going to tell you.
Phytoplankton.
Well, you just mentioned some of them. Algae or that, but the phytoplankton are the plant parts, right? So the zooplankton would be there too, right?
Yeah.
Take advantage of that bloom, an algal bloom. So can you name any zooplankton critters?
No.
Rotifers are some of them.
Rotifers.
Remember rotifer?
I do.
Sure. And
Very
how about
interesting.
ostracods?
You know who works on rotifers?
I don't.
Matt Meselson.
I didn't know that.
Of the Meselson and Stahl experiment.
I know that experiment.
Which showed
N15.
DNA was...
That's right. Meselson works on rotifers? I didn't know that. Good for him. Or her.
Yeah, I'm reading a book called The Red Queen, and
oh yes,
it was an important organism in trying to understand why sexual reproduction is so important as opposed to asexual reproduction.
Okay,
it has characteristics.
For recombination.
It has characteristics which make it very interesting.
Right, but the point is this, though, Vince. Now, if there was a chance of catching Giardia, this would be the time.
From unfiltered New York City water.
Correct, and yet we don't see.
We don't get it. It's diluted, though.
We don't see an increase. So while you were in New York State in the summer, which was even better, you could have scooped up the center of lake water and used that without filtering it at all and been perfectly well off.
I always wondered why reservoirs work and they don't get people sick.
Right.
But when you have a lot of runoff, they do chlorinate them, don't they?
We lightly chlorinate this, but this wouldn't touch this cyst. So you're asking me, wait a minute, I was going back to a point here, and that was that you're camped out, and now you're going to boil some water. You say we don't boil it, we filter it, but let's say we're going to boil it instead, okay? You don't have a filter. You're up in the mountains. You got no filter. And now they sell them, of course, these little ceramic pumps, which are fantastic, but they do clog up. You're right. So boiling them gets the stuff out and makes you can use it again, right? If you boil water at sea level, what is the temperature of the water, Vince?
100 degrees centigrade.
Correct. Now, what if you boil that water at 10,000 feet?
It's probably 95, right?
It's even less than that.
90?
It's so low that you can't kill the cyst stage of Giardia.
Yeah, really?
You can't kill them by boiling it. Not at that altitude.
Would you find 10,000 feet here in New York State?
No, I don't think the highest peak is that high. I think Mount Marcy is the highest peak in New York State, and it's about 6,500 feet.
Yeah. But
if you go out west...
Point well taken. How about my UV pen that I used to take to India, but I never go to India? My wife does.
What does UV radiation interfere with? This is a molecular question now.
RNA synthesis from DNA.
No.
Well, it breaks the DNA.
And then what?
Makes thymine dimers.
Correct.
That blocks transcription.
Well,
basically.
okay. But it can also induce mutations too, right? And that's how that
can
occurs.
induce mutations.
After it repairs, it doesn't repair exactly
can be
the same way.
repaired.
Right. So, okay, so thymine dimers, right? They take advantage of intact DNA, but it has to be replicating in order to have some effect here, right? The cyst stage is a totally resting stage. There's no metabolic activity whatsoever. That's not going to work either, so thank you very much. Next.
Yeah, but when the cysts get activated, then the dimers will be there and it'll kill them, right?
Doesn't work. Sorry.
So they're resistant to UV.
They are.
So when you take the UV pen to India,
yeah, forget it.
What is in the water there that you're worried about? Everything.
No, it might be bacteria and viruses.
Not Giardia, though.
If you knock apart their DNA with it, I mean, you can also break it apart this way too, right? If it's naked DNA.
7 different kinds of genome, by the way.
David Baltimore actually.
He's going to be here in 2 weeks.
Excellent. And that's right. We're working up to the anniversary.
So
to prevent this infection,
your UV pen doesn't work.
Remember, I said avoid contamination with human feces. Good luck on that.
And then we have the river water.
Remember Robert Shope Sr.?
Yes. The world is covered with a thin layer of feces.
Well, he didn't quite say it that way, but that's right.
You said he had a sign over his door.
He used the other word, Vince.
Yeah, but this is a non-explicit podcast. We can't do that.
This is all true. I wouldn't dare denigrate it by dragging it to that level, but you're right.
Little kids wouldn't be able to listen to it then.
They would not, but they know that word better than we do.
Yeah, there's a lot of feces everywhere. But Dickson, when you go fishing, don't you splash some water in your mouth now?
You do, every now and then. Sure you do. Of course. I've been thirsty enough, Vince, and my Gatorade was gone.
You drink the river water?
I took a chance.
Why?
Took a chance.
Because your thirst is so imposing?
Yeah.
Oh, and you're fine, though, right?
I think so. I could have used a little Giardia about a year ago.
I read recently that the idea of getting Giardia from fresh water is really, the risk is so low. It's overblown. And next time I will bring that reference in to show everyone, to show you.
Sure. But the point is that you're out on your own in nature. The best thing to eat if you're up high is snow.
Well, they said in this
melt
book,
snow.
It's actually a book, that camping is okay unless there are a lot of people around. Then you have fecal contamination, and that's where you're going to get the Giardia from.
No, it's beavers. It's the beavers. Beavers.
Okay.
And I've seen them all over the place out west. I mean, there's tons of them all over the place, right? We've stopped trapping them.
Why would we trap them?
When the Hudson's Bay Fur Company went out of business.
Oh, the trapping for furs, yeah.
Forget about it.
Hey, Dickson, we've run out of time.
Hey, that's a shame because we've got 2 more organisms to talk about.
Sounds like we have 2 more episodes.
I believe so.
Is there anything else to say about Giardia?
I'm sure there's a lot more to say about Giardia.
Okay, so if I've gone camping and I come back and within a few weeks I have steatorrhea alternating with constipation, I go to my physician if he or she is smart.
Then to make the diagnosis, I told you if you're going to use the microscope and you see it, you're in luck. But what if you don't see it and you still think it's there? How can you possibly identify it? Okay,
PCR.
Well, that's right. That's one of the methods, and that's a great method because that'll detect remnants of the DNA molecule from the specific pathogen, and you've made the diagnosis. But there's another more interesting technique, I guess, maybe not so more interesting, but you can actually use an antigen capture ELISA test.
Right.
So they've actually injected trophozoites and cysts into goats. And they've collected the antibodies and then used them in a antigen capture ELISA scheme to show that you actually have antigen in your stool. Okay, so you use the stool, but you don't look for the organism. You're looking for the antigen now, which is remnant. Okay, it's very, very large, small amounts, but because you've got a specific goat anti-Giardia antibody stuck on the surface of your 96-well plates, the antigen sticks, and the next thing you know, you use another antibody with horseradish peroxidase, and bingo, there it is.
So then you're diagnosed, you get treated with this drug, cures it, gets rid of your infection?
There are some other, not necessarily. From my readings and from hearsay from various clinicians, I don't know of any drug-resistant Giardia yet.
But so that will cure you and you're fine.
Yeah, but if you happen to miss it because the drug doesn't reach therapeutic levels below the mucosal layer, you just follow it with another round of drug.
What's around? How long?
2 weeks.
So then you can go back a year later and get reinfected?
That's the tragedy of this thing. There's no permanent immunity, so you can get it again.
Well, it's not a tragedy because it's not fatal.
No, but I mean, if you're You don't feel so great when you have this, Vince. There are some mood changes that occur as a result of this, too. It induces temporary depression.
Mm-hmm.
People with protracted diarrhea start imagining that it's caused by something else, like a bowel tumor, or,
Sure.
they go through these machinations of, well, maybe I've got something more serious than Giardia because we haven't made the diagnosis. It's very difficult to make the definitive diagnosis on a single stool sample of anything. We recommend 3 stool samples, 1 day, 2 days, 3 days apart, okay, and send them off to a regional laboratory with expertise in this area. And if you don't find it in 3 stools, they used to recommend take 3 more. So you go through at least 6. Now for you, what if you still don't see it and you don't have any PCR reagents and you don't have any anti-Giardia reagents to do the capture ELISA test? There's one other test.
Mm-hmm.
It's called the string test.
You eat a string and then you pull it out?
How did you know that? How did you know that?
I just guessed.
That's a damn good guess, Vince. So you have this string. It's a braided nylon string.
Mm-hmm. How long?
It has to be from your mouth to your duodenum.
It's about 3 feet.
3.5 to 4 feet. That's right. So what you do is you tape the outside end of it to your cheek at night just before you go to bed. You don't eat anything for din-din. You swallow a glass of water, and down goes the capsule, and it starts to unwind as it dissolves, and eventually it ends up in your duodenum.
Wait, what's taped to your cheek?
The other end of the string. Otherwise, how are you going to catch it and bring it back up again? So the string is attached to your cheek.
And the other part is in a capsule.
Yeah, but the capsule dissolves, releasing the rest of it.
You don't have to swallow the whole string.
You do.
It's in the capsule, though.
It's in the capsule, but it slowly unwinds in your esophagus, and finally, when it's in your stomach, it eventually ends up in your duodenum. And it's pressed up against the perimucosal space. And in fact, it includes everything in the perimucosal space and on the surface of the...
Very clever. Then in the morning, you pull it out.
Bingo.
It must feel good.
Oh, tremendous. Especially that last foot.
But when you've swallowed, you've got something permanently in there. I guess it's so thin that it's not going to affect your breathing, where the flap has to close and all that.
Absolutely not.
So the last you pull out, and then you put it in a tube and give it
And with gloved hands, of course, we always tell the physicians this. On a slide, you express the fluid contained in the interstices of the trihelix.
You actually squeeze it out.
Oh, yeah, and you put a drop of that on the slide. You put a cover slip over it, and in about 90% of even the lightly infected cases, you'll find the trophozoites.
Interesting.
Yep. There's another disease called strongyloides.
I know why they do that.
Sorry?
The trophozoites should be digested away. No, not in the
no. They're in the fluid inside, and it's quickly.
Okay, interesting. What is that called?
The string test. Complicated name for a complicated test.
It's very interesting.
It is. They've gone to great lengths, excuse the pun, to actually make this diagnosis.
Very good.
Right.
You're almost at the level of Alan Dove today.
I'm afraid so. Well, we're making up for his absence. There might be some TWiP listeners that don't know who Alan Dove is, so he's on TWiP and is a Quipster.
Quipster on TWiP.
That's right.
This week in virology.
Yes. So in summary, Giardia has a lot of nuance of its biology that we still don't understand. We're not fully cognizant of its structure as to what it's composed of. We certainly don't know much about its transcriptional behavior. We don't know anything about strain differences with regards to whether it induces a full-blown infection or whether it results in a carrier state. We don't know anything about immunogenetics of susceptible populations versus resistant populations. And we certainly don't know much about preventing it.
Dickson, if you took all the Giardia away from the world, would anyone care? Does it help?
I think the people with Giardia would care a lot.
They would like it, but
they would love it.
Is it any benefit to us? Not really.
Well, I just told you.
So the kids grow better, but not everyone does that.
No, but a subset of those people have actually benefited from having Giardia. So parasites ain't all that bad.
How many different species carry Giardia besides beavers and dogs? Dogs do, right?
Most mammals carry Giardia.
Is this something, if you have a pet dog, you need to worry about?
I don't know if you should worry about it, but they certainly have an option for catching it.
And does the vet know how to cure it?
Yeah, sure. Mm-hmm. It's metronidazole.
So many different animal species carry this, warm-blooded, right?
Yeah.
Every one, would you say? Everything?
Well, I don't think we've looked at every one.
Birds?
Of them. No, not birds.
Why not?
Host range restriction is mammals. And
But
why?
birds,
You're asking why?
birds are warm-blooded, right?
Very warm-blooded. In fact, they're too hot, probably. I can't give you a good reason for that, Vince.
All right.
But I do know what the answer is. The bottom line is that birds don't carry Giardia.
Squirrels?
Oh, yeah.
Chipmunks?
Sure. Bunny rabbits?
Bears?
Bears, absolutely.
Cats?
Sure. Primates, all primates.
Um,
I'm not sure. Insectivores.
Snow leopards?
I don't.
I'm just being silly now.
That's okay.
A lot of animals. Okay.
Yep. So
All right.
that's Giardia. I mean,
That's
Giardia, ancient organism with a rich history in biology, and continues to cause clinical problems throughout the world. And by the way, failure to gain is a big syndrome out there, and Giardia is a big causer of that. So if you're asking what the public health aspect of Giardia is, I can sincerely state that beyond a shadow of a doubt, everybody's convinced that if Giardia went away, and of course the child didn't acquire another diarrheal disease agent, which also caused a failure to thrive syndrome, Giardia is a chronic infection in a lot of people that does cause chronic failure to thrive syndrome in small children, and that disadvantages them to a lot of other things. So getting rid of Giardia would be a great thing to do if we could do it.
Well, we can't. It was just a thought question.
Well, in this country at least, and in most developed countries, we do it through sanitation.
Yeah.
Right? But even there, we have breaches. Like, look, we had 20,000 cases in what you'd consider to be a really well-sanitized country, the United States.
The U.S., we are.
We are. Compared to the most parts of the world, we certainly are. And yet we still can't get rid of it. So I don't think we can ever get rid of anything except smallpox.
All right, Dickson, would you care to answer a few emails?
I'll try.
Our first is from Tom, who lives in Orcas Island, Washington.
Orcas Island? That must be the killer whale island. I like it.
Nice name.
It's a great name.
Dear Mr. Racaniello and Dick. Familiar with you, eh?
I guess.
Thanks for TWiV and TWiP, as both are great shows. Such a give and take of history, information, and humor. Stumbled across TWiP several weeks ago and gave it a try. My only disappointment was there are not many podcasts.
You know, Vince, we get that as a typical complaint.
It's okay. We'll fix it one year.
Maybe we'll do a TWiP a day and then they'll get sick of us.
Thought I would try TWiV, and to my delight found that TWiV was not a new podcast, so there were many hours available for my listening enjoyment. No pressure here, but if you can do more than once a month would be great.
We're going to give it a real shot here. I think once a week we should definitely stick to that schedule, Vince.
Okay, with me, you're just a busy guy.
No, I'm not that busy now. I can do once a week.
No disrespect to Dick in my greeting, but I feel a certain familiarity towards Dick. So the greeting, Mr. Racaniello and Dick, is I happen to pick up some prime specimens of what he spent much of his academic life studying. Some 10 years back, I spent a bit of time bumming around Venezuela, and as they say, the rest is history. Sounds like he got Trichinella.
Maybe.
It was fun listening to the two of you cover this topic in episodes 3 and 4. Thanks for sharing your insight and humor with the rest of us, Tom. Orcas Island.
Our pleasure.
All right. Here's Jim from Virginia. This is for TWiP. An hour-long Radiolab broadcast about famous tumors includes a discussion of the Tasmanian devil tumors.
Oh, yes.
During which the question is asked whether these communicable tumors can be considered a parasite. What do you guys think? The segment is covered in about the first 15 minutes of the program. We'll put a link. It's a program called Famous Tumors. What else do you think would be in that program, Dick, besides this Tasmanian? I bet Peyton Rouse's chicken tumor would be in that one.
If it's not, it should be. That's for sure.
That's for sure.
That's right.
The HeLa cells are also nicely discussed and linked to polio research.
Uh-huh.
The Radiolab guys do good work, just like you dudes.
Hey, I was on Radiolab.
But they have a bigger audience.
I was on Radiolab.
I know you were. We need to have an audience the size of Radiolab.
Well, let's go and ask them how they do it then, because I think they have backing from NPR, by the way. That's a little bit different from us.
So he says, got 10 buckets of peaches off one of our 2 peach trees.
Oh,
Finally,
nice.
After 10 years of trial and error, my lab. Had to use malathion at the end and regret that, but the stuff sure works well.
Yeah, I'm sure it does. Just for a while, though. Keep using it and you'll see.
What happens?
The insects become resistant to it, of course.
So what do you know about these Tasmanian devil facial tumors? Anything?
I know nothing. I don't think they're transmitted by viruses. So
No, they are caused by a clone of malignant cells.
Yes, and then Tasmanian devils are vicious little animals. When they tear apart a carcass, I was in Tasmania, so I can verify this. I actually killed a wallaby by mistake driving at night. And I went back the next day to look for the carcass, and it was gone. How do you think? Where did it go, Vincent?
Someone ate it.
They didn't just eat the carcass. They ate everything. They ate the fur. They ate the bones. They ate everything. And in fact, when a Tasmanian devil dies, it's eaten.
By another Tasmanian devil? And that's how this tumor is transmitted?
Well, I'm not sure, but I know that when there's a carcass, there's a bunch of Tasmanian devils that jump out of the woods and start gnawing away at this thing, and they probably nick each other up in the process. It's a vicious, like a feeding frenzy of piranhas. That a land piranha. I would say that the Tasmanian devil is equivalent to a land piranha. And there's just a frenzy of activity, and then all of a sudden the carcass is gone, and back into the woods they go.
So there's an article here by Carl Zimmer in December 2009 in the New York Times.
Hey, Carl.
And so the scientists discover origin of a cancer in Tasmanian devils.
Yeah.
It's almost extinct, apparently. It's going towards extinction because of this cancer.
I'm afraid so.
It spreads like a plague. So it was reported in Science. Okay, let's see. What does it say?
They could save it, of course, by just taking the Tasmanian devils that are not infected and moving them into zoos throughout the world.
You were just right on. Listen, the cancer, a facial tumor disease, is transmitted when the animals bite one another's faces during fights. Dickson, you know everything.
No, we don't.
It grows rapidly, choking off the animal's mouth and spreading to other organs, has wiped out 60% of all devils since it was first discovered in 1996.
Yes. By the way, the only ones that are going to survive this are the Tasmanian angels, because they don't bite each other's face.
Chromosomes of these cells look less like those in the normal cells and more like those growing in the tumors. In 2007, they compared the DNA from sick and healthy animals, and they found the cancer cells shared distinctive genetic markers not found in the animals themselves. So now...
This has been transmitted from animal to animal.
It has been. So in the cells, they find a set of genes normally active only in Schwann cells.
Interesting.
They argue that a single Schwann cell in a single animal was the progenitor of all the devil facial tumor cells. Wow. How about that?
Wow.
So somehow a Schwann cell went wild and took over. But that's interesting that it gets transmitted without being rejected by the immune response.
Yeah, that's right.
Suggests that these animals are genetically very, very similar, doesn't it?
Well, look where they live. They live on a single island.
Right.
And there's no other species population anywhere else. And they make lots of noise at night also. They're not nasty little animals. But I still feel badly for them. I wouldn't like to see them to go away.
The next email is from Heather.
Okay, Heather.
I am a third-year medical student here at Columbia.
Cool.
Dr. Despommier actually interviewed me when I applied and had a lot to do with my decision to choose this school.
Okay, Heather.
And then he told us about TWiP during a parasitology small group session.
It's true.
Last fall, and had a lot to do with my listening to this show when I should be studying for exams.
Heather, go back to studying, please.
I thought you would get a few seconds of amusement from a medical student's interpretation of one of Dr. Despommier's lectures. Please see the attached PDF with a portion of my notes from the first parasitology lecture he gave last fall. Here you go, Dickson.
This is my last lecture ever, by the way.
Toxocaracanus and Toxocaracati. Now, I don't know what this is. We haven't talked about Toxocara.
Nope. There are ascarids that live inside dogs.
So here we have,
cats.
Her notes are scribbled on this slide, associated with people who have a lot of pets, breed dogs, go to parks with dog runs.
Yep.
So we have dogs shedding these worms, are they?
Eggs.
And it's going down here. You're swallowing it, and it goes in the intestine.
Yep.
And she writes, visceral larvae migrates, ocular larvae migrans.
Yep.
But then at the bottom, in quotes, holy S, I'm not in a dog.
I might have said that in passing. No pun intended.
Then it says, it's going into the blood. Now where do I go?
Right.
And then finally, wherever in the eye. So it can go anywhere. Huh? So if it's not in a dog, it causes bad news.
It gets lost once it gets inside a human.
I think we should put this on the website.
Sure.
Is that okay, Heather? Well, Heather's not listening.
Yes, she is listening, actually. Heather, you take good notes, I must say that.
And finally, the last one is from Marshall, who writes, I just wanted to make sure you saw this. Apparently, a mosquito has been genetically altered to be resistant to malaria.
I do know this.
I haven't had a chance to read the PLOS article, but maybe you'll find this interesting. Is this interesting, Dickson Despommier?
It's fascinating. Who wrote the article?
Researchers from the University of Arizona have introduced a gene that affects the insect's gut, meaning the malaria parasite cannot develop.
That's what we've been looking for a long time.
Professor Michael Riehle, R-I-E-H-L-E, from the University of Arizona. And they want to release these, of course.
There's some really interesting reasons why there will be arguments over that. Because malarial transmission might be stopped, but the same mosquito can transmit lots of other things, too, including encephalitis.
They completely block replication of malaria in the mosquito.
But they could transmit other things, though, Vince. So it's essentially like saying malaria is the worst of our worries, so let's try to prevent that. But I'm probably going to increase the rate of transmission for, let's say, filaria parasites or for encephalitis parasites. And you have to make a choice.
Well, you'd have to check all those, right? That would be difficult.
Yeah, you can't make them resistant to everything.
So are you saying that we can't release these types?
I didn't say that.
Because it would be...
I will say that if they take this mosquito in mass to an area of high malaria infection, they'll have to get a lot of permission slips from the government in order to carry out this release.
All right, Dickson, that will do it. You can find TWiP on iTunes, of course. Yep. You can find it on microbeworld.org/TWiP. You can find it at sciencepodcasters.org and promednetwork.com. And if you're a new listener, go on over to iTunes, leave a comment. It helps us to stay on the front page of the medicine podcast section. If you have questions and comments, we love to hear them.
We do.
Send them to twiп@twiv.tv. Another wonderful session, Professor. Thank you.
Welcome, Vince.
Good to see you again.
Same here.
And I look forward to many TWiPs and TWIVs.
Indeed.
You want to say goodbye, Dickson?
Sure.
You know, Dickson Despommier can be found at medicalecology.org. Where else? Verticalfarm.com.
Trichinella.org.
And trichinella.org. You're going to start another website? That's
well,
enough?
No, eventually I'd like to start one on urban agriculture, but I haven't yet decided how to do that.
Urban agriculture.
Yep.
So that is growing things in cities.
That's correct. Yep, it's a big and up and coming field, and I think it needs a wider audience. And we'd like to coordinate the information and put it all in one place. So
I'll bet that would be a very popular site.
I think so.
You ought to start a podcast too.
You think?
Yeah.
Would you help me, Vince?
Oh, of course, Dickson.
Okay.
But I don't know anything about I can record and listen, but and ask stupid questions.
You could ask great questions. You don't ask stupid questions. You ask great questions, Vince. I mean, I get accused of asking great questions in This Week in Virology because I'm not a virologist, but I'm a biologist. So if you have a biological focus to your life, and if it's a biological subject, it doesn't matter what the subject is, you can still ask relevant questions. I
think your questions for TWiV are wonderful.
Well, somewhere and somewhere.
They're all fine.
Yep.
You have been listening to This Week in Parasitism. Thanks for joining us. We'll be back soon. Another TWiP
is parasitic.